everyone. This is Lecture 7, which will cover the brain organization and function. In this lecture, our major objectives are to identify the major parts of the brain, identify the major structures of the brain, such as the meninges or the skull, cerebrospinal fluid, and the blood-brain barrier. We'll also talk about the function and circulation of cerebrospinal fluid. We'll see how the distribution of gray and white matter is different in the brain, and the major lobes, fissures, sulci, gyri, and functional areas of the cerebral cortex. We'll also mention the lateralization of the cerebral hemispheres. Lastly, we'll discuss the functions of the motor, association, sensory, and limbic areas of the cerebrum. So let's get started. The brain organization and function. Well, we're all we know from SCB 203 that the brain is located in the cranial cavity and controls most of the body's functions. Some basic facts of the brain. It's composed mostly of nervous tissue. It has internal cavities, which we mentioned in earlier lectures, called the ventricles, that contain the cerebral spinal fluid. It weighs about 1,200 to 1,500 grams and receives about 20% of blood flow. As you might expect, such a vital organ has a large amount of the cardiac output. We'll also see that when we discuss the urinary system. The divisions of the brain. We can divide the brain into four basic subregions: the cerebrum, the diencephalon, cerebellum, and the brainstem. Now, if you go on the internet, you're going to find various other ways of dividing the brain up, both structurally and functionally. But we're going to go by this based on your lecture text. The cerebrum. This is the largest part of the brain and is composed of two cerebral hemispheres. As you can see in this figure, we see the left cerebral hemisphere. And notice that there are elevations as well as depressions, which we'll discuss later. The cerebrum is important for learning, memory, personality, cognition, language, and conscience. So these are all characteristics that we attribute to us humans that distinguish us from most other organisms. It also has major roles in sensation and movement. The diencephalon. This is the central core beneath the cerebral hemispheres. If we look at it, it's sort of like a little peach pit in the very center of the brain. Its central localization is reasonable based on its functions, as it's important for processing, integrating, and relaying information to different parts of the brain. So again, structure reflects function. It also has homeostatic functions such as regulation of movement and biological rhythms. The third part of the brain, which is probably the second largest, is called the cerebellum. And it's separated to right and left hemispheres, much like the cerebrum. And you can see the cerebellum in this kind of pinkish area in the inferior part of the brain. It has important functions in planning and coordination of movement. So other organisms may have a smaller cerebrum, but oftentimes they may have a fairly large cerebellum in proportion to the brain due to its involvement in the control of movement. Finally, we have the brainstem, which is sort of like a little stick that's sticking out from the bottom of the brain that continues with the spinal cord. And it's the oldest part of the brain, and as mentioned, it connects the brain and spinal cord. Since it is the oldest part of the brain, it has the basic functions of controlling body functions. Okay, so here in purple is the brainstem, and you can see it continues on with the spinal cord. So it controls the basic involuntary homeostatic functions, such as certain reflexes, monitoring movement, integrating and relaying information. Now, we mentioned the gray and white matter in the spinal cord. There's also gray and white matter in the brain as well, in the cerebrum. But as we will see, that the gray and white matter are opposite to that of the spinal cord. In the cerebrum, the gray matter is outside to the white matter. Remember, in the spinal cord, it was the white matter that surrounded the butterfly-shaped gray matter. Two other terms we mentioned in previous lectures are tracts and nuclei. And these are going to be important means of communication within the cerebrum. And remember, tracts are bundles of white matter that communicates with nuclei, which are the cell bodies, dendrites, and non-myelinated axons in the gray matter. We also have what are called primary brain regions. These are developmental regions that include the forebrain, midbrain, and hindbrain that develop into the mature brain. For now, we're just going to mention them, as you might experience these terms in your readings. So the three primary regions are the forebrain, the midbrain, and the hindbrain, and they're named because of their relative location in the brain. So anterior is the forebrain, and this becomes the cerebrum and diencephalon. The midbrain becomes part of the brainstem, and the hindbrain becomes the cerebellum, pons, and medulla oblongata, another part of the brainstem. Now in the figure, we see these three regions and how they develop into the mature structures of the brain. For now, don't worry about these secondary brain vesicles, as if you take a more advanced neuroanatomy course, you'll learn about these developmental regions. Protection of the brain. Okay. We learned from SCB 203 that you have skull bones that will surround the brain to protect it. So we won't go over that in this lecture. However, there are other structures that will protect the brain, such as the cranial meninges, the cerebral spinal fluid, and the blood-brain barrier. The cranial meninges is similar to that of the spinal meninges, and it has the same three basic layers: the outermost Dura Mater, a middle Arachnoid Mater, and a deep Pia Mater. The Dura Mater is the outermost and the thickest layer, and superficial is the epidural space that contains blood vessels. So in this figure shows the Dura Mater. And as mentioned before, the epidural space contains blood vessels. The subdural space contains serous fluid and veins. And so here is showing the subdural space here. There are also dural sinuses that will help drain CSF as well as deoxygenated blood. And so here's an example of one dural sinus that will reabsorb the cerebral spinal fluid. The Arachnoid Mater is innermost and covers the very surface of the brain. It's very thin, and it contains cerebral spinal fluid and blood vessels. Specialized granulations return the CSF to the bloodstream. And we see in this figure here, we can see the Arachnoid Mater, and beneath it the subarachnoid space. And here is showing the example of the arachnoid granulations. As mentioned before, the Pia Mater covers the immediate surface of the brain. In the lab, you would see an example of the sheep's brain, and you'll notice that the Pia Mater is almost continuous with the surface of the brain. And as you might expect, structure follows function. And being so thin, it is permeable to substances in the extracellular fluid and the cerebrospinal fluid. So here in this figure showing the Pia Mater as the innermost layer, immediately covering the contours of the surface of the brain. Now we mentioned that the brain is hollow. It has spaces called ventricles. And these ventricles are cavities that contain the cerebrospinal fluid. And actually there are also areas that produce the cerebrospinal fluid. And these ventricles are continuous with one another, as well as the central canal of the spinal cord that we mentioned in a previous lecture. There are 4 ventricles. The first 2 are the right and left lateral ventricles. There's a 3rd ventricle, and there's a 4th ventricle. And this figure shows all 4 ventricles. So in this lateral view, we see the right and left lateral ventricles. We see the 3rd ventricle, and then we see the 4th ventricle. Okay, in this anterior view, we see the lateral ventricles. Here's the 3rd ventricle, and here's the 4th ventricle. In the laboratory, you will see a gray model that depicts just the ventricles alone, which you will probably see in a PowerPoint. Now, 2 other structures of the ventricles is what connects each of the chambers together. The first is the interventricular foramen, and this connects the 3rd ventricle with the lateral ventricles. And also the cerebral aqueduct connects the 3rd ventricle with the 4th ventricle. And then the 4th ventricle is continuous with the central canal of the spinal cord, which we mentioned in a previous lecture on the spinal cord. Let's talk a little bit more about the cerebrospinal fluid. We mentioned it several times already, but we haven't really described its composition. It's a clear, colorless liquid similar to plasma. Of course, there are no blood cells present. It has various functions. One, it protects by cushioning the brain. It helps maintain a constant temperature so your brain doesn't overheat or get too cold. It helps remove waste. And this is an important area of research now, that the reason why we need to sleep is that while we're sleeping, the movement of cerebrospinal fluid removes all the waste and toxins during the night. And some research indicates that failure to get sufficient sleep may lead to neural disorders later in life. Functions of the cerebrospinal also, it increases the buoyancy of the brain. So remember, the brain and spinal cord are actually buoyant in the cerebrospinal fluid. Now, a region in each of the ventricles is called the choroid plexus. And it is the choroid plexus which secretes the cerebrospinal fluid. And the choroid plexus occurs where the fenestrated capillaries contact the ependymal cells. Remember, the ependymal cells are a type of neuroglial cell that produces the cerebrospinal fluid. So this figure is a kind of a complicated figure, but we just will simplify it a little bit. A region of the ventricles is called the choroid plexus that produces the cerebrospinal fluid. And if we amplify that region, we can see here's a capillary, and here's the cerebrospinal fluid, and the ependymal cells that are producing the cerebrospinal fluid. And the cerebrospinal fluid circulates throughout the brain with little cilia. So it's constantly moving, even while we're sleeping. There's about 150 milliliters of cerebrospinal fluid that circulates through the brain and spinal cord. However, there's about 800 milliliters of cerebrospinal fluid that is produced each day, which means that cerebrospinal fluid is actively reabsorbed in order to maintain this constant 150 milliliters. Now, problems in the reabsorption of the cerebrospinal fluid may cause very dramatic problems. One example would be hydrocephalus, which is swelling of the brain. If there's blockage of the reabsorption of the cerebrospinal fluid, and this will increase intracranial pressure. And what they need to do is probably remove some of the skull bones to allow room for the brain to swell until it can be the volume can be decreased. Another protective function or protective structure is the blood-brain barrier. As its name, the blood is separated from the brain. So the cerebrospinal fluid and the brain extracellular fluid has a barrier from the blood. How does it do this? Well, the blood-brain barrier consists of an endothelium of capillaries and its basal lamina with astrocytes. These endothelial cells of capillaries are very unique because they have more tight junctions than your traditional body endothelial cells. And with tighter junctions, it will limit the endocytosis and exocytosis across the membrane. So, in this figure here is a comparison of a typical capillary in the body, external to the brain, and we can see that they're leaky to allow things to pass through. However, in the endothelial cells of the capillaries in the brain, we see they have much tighter junctions that will prevent movement of fluids easily across the membrane. Furthermore, astrocytes, another type of neuroglial cell which we mentioned earlier, has these foot processes that extend and sort of like help seal and form a clamp around the capillaries. In this fashion, the endothelial cells of the tight junctions and the astrocytes produce the blood-brain barrier. Now let's talk about each of the different regions of the brain. The cerebrum, for example, is composed of the cerebral hemispheres and contains the lateral ventricles, which we just mentioned. In a superior view, we nicely see the two cerebral hemispheres. Here's the right cerebral hemisphere, and here's the left cerebral hemisphere. Again, notice the elevations, indentations on the surface. It is not a smooth surface. If you remember structure and function, this will increase the surface area to allow more neurons in the finite space of the cranium. And they have names. The sulci are the shallow grooves on the surface. A single one is called a sulcus. Gyri are the elevated ridges, and a single one is called a gyrus. And this diagram shows rather small, but we see the elevations of the gyrus and a single depression as a sulcus. In addition to these, there are other surface structures of the cerebrum. For example, fissures are deeper grooves. One such example of fissure is the longitudinal fissure that separates the right and left hemispheres. In this figure, we see again the right cerebral hemisphere, the left cerebral hemisphere, but along the middle of it is the longitudinal fissure. There are various lobes of the cerebrum. There are 5 lobes, 4 of which can be seen on the surface of the cerebrum, and 1 which is deeper. And the first 4 are named from the skull bones that you learned in SCB 203. And in the right here, I have a brief function of each. The frontal lobe is for planning and executing movement and complex mental functions such as behavior, conscience, and personality. The parietal lobe is for processing and integrating sensory information and function and attention. The temporal lobe is for hearing, language, memory, and emotions. And the occipital lobe is for vision. The last one, insula, is the one that cannot be seen from the surface of the brain, and it's involved in taste and visceral organs. Let's talk a little bit more about the frontal lobes. So these are the anterior-most lobe of each cerebral hemisphere, and its posterior boundary is called the central sulcus. The gyrus that's anterior to the central sulcus and still part of the frontal lobe is called the precentral gyrus. So in this figure here, we see the frontal lobe, okay, and we see the central sulcus that's at the posterior boundary of the frontal lobe. Here again, here's the central sulcus. Now, the gyrus immediately anterior to the central sulcus is the precentral gyrus. Over here is the precentral gyrus, and it has important functions which we'll talk in a little bit. The parietal lobes are posterior to the frontal lobes, and these are important for processing and integrating. Since they're posterior to the frontal lobe, they're posterior to the central sulcus. And the gyrus that's immediately posterior to the central sulcus, as you might expect, is called the postcentral gyrus. Okay, so here's the frontal lobe, here's the central sulcus, and posterior to that is the parietal lobe. Okay, so here's the parietal lobe, here's the central sulcus, and the gyrus immediately posterior to it and part of the parietal lobe is the postcentral gyrus. So we have the frontal lobe, the precentral gyrus, the central sulcus, postcentral gyrus, which is part of the parietal lobe. The temporal lobes, as you might expect, is on the lateral surface and separated from the frontal and parietal lobes by the lateral fissure. So let's take a look at this. Here's the temporal lobe, and you can see the lateral fissure here that's going to separate the temporal lobe from the frontal and parietal lobe. The occipital lobes are posterior to each cerebral hemisphere and separated from the parietal lobes by the parietal occipital sulcus. In this figure, we see the occipital lobe, and it's separated from the parietal lobe by the parietal occipital sulcus. The last lobe of the cerebrum is called the insula, and it's the only one that cannot be seen from the surface of the brain. So it's deep to the frontal, parietal, and temporal lobes. In order to see it, you have to pry apart the superficial lobes to see the insula. Now, as we mentioned earlier, the gray matter in the cerebrum is superficial, with the white matter deeper. So the cerebral cortex contains the cerebral gray matter, and in this, the majority of the cerebral cortex in humans is the neocortex. Neo meaning new or recent, and is involved in the conscious processes such as planning, movement, interpreting sensory information, and complex higher functions. So these are all recent evolutionary advances of the cerebral cortex that we attribute to us humans. Functional areas of the neocortex. There's the primary motor cortex, the primary sensory cortices, and various association areas. Let's look at the primary motor cortex. The primary motor cortex is located in the precentral gyrus of the frontal lobe, which we just previously mentioned, and it's involved in the planning, guidance, coordination, and execution of movement. So in this figure, which is a little bit more colorful than the one we showed before, we see the frontal lobe, and we have in red here is the precentral gyrus, which functions as the primary motor cortex. We also have various sensory cortices, And these various sensory cortices have functional functions in touch, vision, hearing, smell, taste, equilibrium, the complex receptors, as well as also what we call the general receptors, such as temperature, touch, vibration, pressure, stretch, and joint position. And these are called the somatosensory areas. So the primary somatosensory area, or primary sensory cortex, is located in the postcentral gyrus of the parietal lobe. So in this picture here, we see the central sulcus, and immediately posterior to it is the postcentral gyrus, which contains the primary somatosensory cortex in blue. Now the visual area, or the primary visual cortex, is located in the posterior lobe, in its posterior end. So in this figure here, we see the primary visual cortex, which is kind of interesting since the eyes are more anterior to the brain. The primary auditory cortex, as you might anticipate, is in the temporal lobe. And it's located here in the temporal lobe, the superior portion of the temporal lobe here. There are various language areas. Two of the most important ones are Broca's area and Wernicke's area. And they may seem very similar in function, but if you read this carefully, they have very distinct functions. Broca's area is for speech sounds to produce language, whereas Wernicke's area is the integrative speech area to understand language. Damage to one or the other of these can have very dramatic effects. For example, if Broca's area is damaged, a person won't be able to produce the sounds for language, even though they might be able to understand language. Likewise, if Wernicke's area is damaged, a person could be able to produce the sounds of language, but won't be able to understand them. And these are located here. Here's Wernicke's area, and here's Broca's area. Now, another region of the cortex is called the prefrontal cortex, which composes most of the frontal lobe. And it's important for functions for modulating behavior, personality, learning, memory, and psychological state. There have been cases where people have damaged the prefrontal cortex through an accident, and they're perfectly normal. However, their personality hasn't changed due to the accident. So here is showing the prefrontal cortex, which is most of the anterior part of the frontal lobe. There are also a number of parietal and temporal association areas that compose each of these lobes. And these are going to be involved in the integration of sensory information, language, maintaining attention, recognition, and spatial awareness. And so they're located here, the parietal association cortex and the parietal lobe, and the temporal association cortex and the temporal lobe. And finally, here's a table from your lecture text that covers everything we have, summarizes everything we've covered thus far. One of the internal regions of the brain is called the limbic system, which includes both gray and white matter. There are many subregions of the limbic system, but we're going to only identify a few of them. One of these is called the hippocampus, and it's connected to a region called the fornix. And so here's the hippocampus. So it's like a little horn here that encircles the diencephalon. The hippocampus has a role in memory and learning. So damage to the hippocampus, you may have, effects in your memory. Another region is the amygdala. The amygdala is just anterior to the hippocampus, so they're very tips of these horns. Here's the left one, and here's the right one. And these function in expression of emotion, especially fear.